Hello everyone and welcome to Physiology for Engineers with Professor Gonzalez-Fernandez at Lehigh University. Today we are going to talk about the glomerular filtration, which happens in the kidneys. So the kidneys are these two bean-shaped organs that are here in our lower back, and these organs are pretty important because they are going to filter all the waste products in our body, and then they are going to excrete those products into the urine. So here we have a little schematic of the kidneys microscopically. So here we have the two being kidneys, and then these kidneys are very important because they are going to excrete the urine through the ureters here. So these are the ureters. And they are going to bring this urine from the kidneys through the ureters into the bladder. And finally, we are going to urinate and expulse all this urine through here, the urethra. So also, because the kidneys have to form the urine, have to filter all the blood in our body, it's really important, and they are highly vascularized. So here we have arteries and we also have veins. So the main artery is the aorta. So the aorta is going to be divided into the renal or kidney arteries, which are going to bring blood into the kidneys to be filtered. And we also have veins. So the most important here is the inferior vena cava. So this inferior vena cava is also going to be divided to go to the individual kidneys, and these little veins that come from the vena cava are called the renal veins or kidney veins. So now in this class today we are going to look more deeper into this and look at all the processes and all the forces that are going to guide the filtration process. The filtration unit, where all our blood is going to get filtered and we are going to form all the urine and we are going to get rid of the waste products, is the nephron. And the most important part, the functional part of the nephron here, is this part in the schematic, which is formed by the glomerulus, and it's also formed by the Bowman capsule. So this is the glomerulus. And around the glomerulus, we have the Bowman capsule. Okay. So here in the glomerulus is part of the kidney arterioles. So the blood is going to get into the glomerulus here through the afferent arteriole. So it's called afferent because the blood comes into the glomerulus through the afferent arteriole. And then the blood is going to get filtered here at the glomerulus and with the Bowman capsule, and then it's going to come out here through the efferent arteriole. Just in case you cannot see that, the efferent arteriole. So it's called efferent because it's going to expulse all the blood from the glomerulus back into the general circulation. So the most important process that happens here in the nephron is the process of excretion. So this our blood is going to get filtered here, and we are going to form all basically the urine. Well, like the urine will get formed as well with all the process of absorption and reabsorption that is going to happen in the tubules. But here we're going to form like the excretion. Okay. And the excretion, let's put it here, which is all the products that we excrete from the blood, is going to be determined by other two processes. So the first process is the process of filtration. And there's going to be another process which is going to determine the excretion, which is the process of absorption. Okay. So we have that excretion is equal to the filtration minus the absorption. And this is how the volume of urine that we are going to excrete from our body is formed, and how the different filtration and absorption of the glomerulus are going to determine this volume. So we are going to look a bit deeper at the different physical forces that mediate the processes of filtration. And we are going to go back to these forces that we have already seen when we were talking about the capillaries. So these forces are the Starling forces. So these are very similar to the different forces that we saw when we were talking about the capillaries and the vascular system, but there are specific things that we need to remember about this. So we have here like a different schematic of this. So this is like a schematic to simplify what we can see here. So here the blood enters into the glomerulus. So here we have the glomerulus, and here we have the Bowman capsule. So the blood enters into the glomerulus through the afferent arteriole, so AA, and it goes through here, and then the blood leaves here the glomerulus through the efferent arteriole. Okay, so these are important concepts that we need to remember. So also in the Bowman capsule we have this specific cell type, which are called the podocytes. So these podocytes are specialized cells that are going to mediate in this process of filtration. And they are going to form this barrier here between the Bowman capsule and also the endothelium in the glomerulus. So let's go for the starting forces. So the first starting force that we have here is the hydrostatic pressure of the glomerulus. So is the pressure here of the blood at the glomerulus, and this is a force that is going for filtration. So it goes here. So here PG, okay? Take this out. The second force that we have is the colloidosmotic pressure of the glomerulus, and this is going to be a force for filtration. Sorry, for absorption. So it's going against filtration. Okay. The colloidosmotic force is due to the protein content. So if the blood has a lot of protein and we are losing a lot of water because of the filtration, then this force, the colloidosmotic force, is going to bring back water from the Bowman capsule into the artery because the protein concentration here into the glomerulus is really high. Then we also have the same forces at the Bowman capsule. So we have the hydrostatic pressure at the Bowman capsule, and the hydrostatic pressure of the Bowman capsule is a force against filtration, is a force for absorption. And we also have the colloidosmotic force at the Bowman capsule, which is a force that it's going for filtration against absorption. So we can use these four forces to determine the glomerular filtration rate. So we can say that the glomerular filtration rate, so let's see if you can all see here. So glomerular filtration rate is equal to So we have here the renal coefficient or the kidney coefficient, which is like a constant, and that only changes if we have a change here of the properties of the barrier in the Bowman capsule. So it's usually the same unless there is like a pathology, like an inflammation of the nephron, for example. So we have this coefficient that is constant. This is going to be multiplied by the hydrostatic pressure at the glomerulus plus the other force that is for filtration, which is the colloidosmotic force at the Bowman capsule, minus the hydrostatic pressure at the Bowman capsule. That again, this is a force that is for absorption, it's against filtration, that's why it's negative. And then minus the colloidosmotic pressure at the glomerulus, that is a force against filtration, and it depends on the protein content. Okay, so this is the formula that is going to determine the filtration process and the rate of filtration of the blood here at the glomerulus. So as I said, the K is the coefficient, and this depends on the barrier here that separates the glomerulus and the Bowman capsule, and it's usually constant. We only have a modification of that if we have a pathology or this membrane here is disrupted for whatever reason. Then PG, so the hydrostatic pressure at the glomerulus is usually around 60 millimeters of mercury. So always millimeters of mercury for pressure. Then we have here the colloidosmotic pressure of the glomerulus, that is around 30. Then we have the hydrostatic pressure of the Bowman capsule, that is going to be around 20. And finally, we have the colloidosmotic pressure at the Bowman capsule, that is usually 0. And it's important to understand why this is 0. So here at the glomerulus, we are only going to have the filtration of water and salt. So there is not going to be filtration of protein. So this is really important, right? Because all the protein will be here at the blood vessel, will be here at the glomerulus. They wouldn't be protein if everything's okay and like the conditions are physiological. There shouldn't be any protein here at the Bowman capsule because the protein is just not getting through the barrier. So because there is no protein at the Bowman capsule, the colloidosmotic pressure there is equal 0. So this is something that we can take out of the equation here, okay? Cool. So now we are going to look a bit more into these forces and how how we can represent them graphically and how they interact with each other.